Welcome. Welcome to the panel, who I'll introduce shortly, and welcome to all of our guests and friends who are joining us today for this. This first time we've done this. We're calling this Compelling Conversations. This is an idea to create something of an intimate opportunity for partners, alumni, supporters of the school to eavesdrop and then participate a little bit in a conversation with experts, people who are really doing compelling and important work. And it's unscripted. We're just going to be talking to each other, but we hope this is interesting. Eventually, we're going to do this on a bi-weekly basis and invite folks in from lots of different areas. Usually, it'll be faculty from our school, but perhaps joined by alumni who are doing interesting things, or partners from around the campus, business leaders, etc. So that's the general idea. For our first conversation, I'm thrilled to have three experts who are working at the forefront of COVID-19 diagnostics. We've got John Gerace, who's an alum of chemistry and also biosci. He's the president of DiaSorin Molecular, which you'll hear a bit about later. Reg Penner, who's a Chancellor's professor of chemistry, and Greg Weiss, who's a professor of chemistry. And Reg and Greg have co-founded a company called Tech that you'll learn a little bit about later as well. And again, they're also working on diagnostics, coming at it from a different way. And I think the three of these folks together is going to provide perspective. So my name is James Bullock. I'm the Dean of the School of Physical Sciences, if I haven't had a chance to meet you yet. I am just an astronomer, a lowly astronomer, so I will do my best to keep up with these chemists as they talk, and I'll try to keep the conversation ironically down to earth, even though I spend a lot of time looking up. With that, let me just briefly introduce these folks. All three of them are esteemed and have done a lot of work in this area and have interesting trajectories, and maybe we'll get into that too with the conversation. Reg Penner is a Chancellor Professor of Chemistry. He's been at UCI for over 30 years. He got his PhD at Texas A&M and he became faculty here in 1990. He works on photonics and energy storage and a lot of different things as an electrochemist. And he's also, as I mentioned, been moving towards diagnostics. And we'll hear a bit more about that as we get introduced. Greg Weiss is a professor of chemistry and molecular biology and biochemistry here at UCI. He's been here for about 20 years. He has an undergraduate degree from UC Berkeley, a PhD from Harvard, and he works on atoms, bonds, molecules, things that make life possible. And again, has been collaborating with Reg for a number of years now on one of these sort of magical collaborations that can happen at an interactive place like UCI. And finally, John Gerace is president, as I said, of DiaSorin Molecular, and he's worked on molecular diagnostics since 1993. Very proud to say he's an alum of UCI with degrees in chemistry and from biosci. And his trajectory is a bit different. I think he thought he was going to be going to graduate school, but ended up getting an MBA and started running a lot of companies. And so he's worked in a number of different places, Beckman Coulter, Freedom Meritech, probably too many to go into, but we can get into that later in the conversation. But he's currently CEO of DiaSorin Molecular, and this is a place that manufactures and distributes lots of diagnostic stuff. One of the global leaders in this area, and as we'll soon find out, is at the forefront of both the vaccine, of the virus detection and also antibody detection efforts. So with that, I'd like to start the conversation, and maybe I'll ask John, how did you get to where you are now, and what is it like to be working kind of at the forefront of just your perspective of what's happening in the world right now with this COVID-19 issue?
Well, thank you for having me. I really appreciate it. It's great to be on this panel. And how I got where I'm at right now, let's try to spare of the long, 30-year history. But, I started off when I went to UCI, I had a lot of interest in cooking, actually, in the kitchen, and that's what attracted me to chemistry. My quest was to find a preservative for my guacamole dip. I had the best guacamole dip in the world. And I knew that it turned brown really quick. You know, you put the seed in it, you put lemon juice, but I was intrigued with other ways of preserving guacamole dip. And that's what brought me to a chemistry major at UCI. And then as we all are really influenced by our professors, I had one professor in particular that really inspired me. That was David Brandt in physical chemistry. He was really an inspiration. And so I've always been interested in the business of science, from guacamole dip to, you know, ways of making life easier through chemistry, right? So it led me to doing research with Allergan for about 5 years. I have 4 patents there on silicone, different types of polymer chemistry. And then I wanted to do more of the commercial marketing end. So I ended up getting my MBA and working at Beckman Coulter for a little over 15 years. So I started getting involved in PCR, the polymerase chain reaction. So again, a Nobel Prize given that year to Kary Mullis, who was a chemist. And I've been involved with some form of either automating it or synthesizing primers or sequencing since that time, so for almost 30 years. I ended up running the PCR business for a market leader in the industry, Applied Biosystems, so the instruments and reagents associated with PCR there, and have always been involved in some form of detection kit, either for research use or for forensics or for food safety. So I was involved in the horse meat scandal, horse meat being contaminated in hamburger meat a while back. And then, you know, recently just, you know, been involved in all of the infectious disease outbreaks stemming from, you know, from the first SARS, MERS, Zika, H1N1, you know, Ebola. So been around the block in that area. So that's basically how I got where I'm at right now and what I do.
That's great. Thanks, Sean. And we'll return to, you know, I think we're going to start with a little background and then we'll return to the topic at hand in a little bit. Let me just say before I move on to Greg, we're going to have some time for Q&A later at around 11:45. We'll start taking questions from the question box. So, you know, hold on and I'll note that time when we're ready to open that up for Q&A. So Greg, give us a little, your trajectory very quickly and how you got into doing diagnostic stuff.
Sure. So first, thanks again, James. This is really exciting for us to chat with this group today. I think my trajectory for interest in diagnostics and this interface of chemistry and biology started when I was a postdoc at Genentech. I was really interested in biotech companies and starting biotech companies. But I was, as a postdoc, which is sort of an exalted intern, you really don't get to do the cool stuff of inventing whole new technologies. And at the same time, I really miss standing in front of a classroom. I love talking about organic chemistry. I love running to work in the morning to see my students and hear the cool things that they did last night. So I really miss that. And you know, at the moment that I was dropping envelopes into the mail, because back in those days, that's how searches for jobs, I made that decision to head to academia, and I was really lucky to land here at UCI, where I've been ever since. But the trajectory to getting towards this COVID-19 test, which is what Reg and I are working on, started when the two of us met at UCI, and almost immediately we became pretty close friends and started talking science. And a tragedy struck us then. In about 2004, 2005, Reg's mother passed away, and my father had passed away also from cancer. So both of our parents passed away from cancer. And we just looked at each other one day and we said, you know, we should do something about this. We can do something that would help stop this disease in its tracks if we can do diagnostics on a much more frequent basis, on a much simpler basis, more quickly, and make it more routine. And so that's what we started doing. We've been collaborating ever since. And then this COVID pandemic struck. And like everyone, I was hunkered down at home. Reg and I actually threw ourselves into the original SARS-1 effort many years ago. And we spent a grueling couple of weeks working on this thing. And honestly, it was the most painful episode because we spent a ton of money working on this. None of the money we were able to get back. And at the same time, we had no chance of getting any samples out of China and out of these places where the disease hit. So I thought, oh no, here we go again. last thing I want to do is get involved in this. So I'm hunkered down at home thinking this is just going to, we're going to ride this one out. There's companies like DiaSorin, John's company, they're going to work on this. They're going to solve this. But then after a week or so of doing that, I started to get really frustrated because I was looking at the technologies that were coming out and I realized no one was bringing to bear the type of stuff that Reg and I do. And so Reg and I, shot some emails back and forth. And before you knew it, we were petitioning to you, James, to reopen our labs. And I was really glad that the Dean's office let us do that. So our labs are now open just for the COVID-19 research. And that's what we're going to talk about.
Yeah, so let me move on. I'll just mention to everyone in case the folks on the line don't know, most of the research on campus is shut down, but we have approved a lot of in-person work for folks who are doing, crucial frontline research on the problem itself. So like Reg and Greg are working on diagnostics for COVID-19. We have other folks in the school who are doing work on masks and doing work on antiviral treatments. And so that kind of stuff is going in a careful way, but it has been turned on. And so it's been empowering a little bit to sort of sit in the dean's office and just be aware and be able to sort of facilitate some kind of response from our end. And I have to say the chemistry department, which is represented here, and I've put it up behind me here just to sort of showcase the excellence, has just been all in, all hands on deck. So many people are turning towards using their own talents and expertise to try to make a difference here. So Reg, let me ask you from your perspective. I mean, very quickly, you have this illustrious career here, and you've worked on all kinds of interesting things in photochemistry, including energy storage and other things along those lines. And now you're doing this. So from your perspective, how did you get here, and what's your perspective on what you guys are trying to do for the antibody testing?
Thanks for using the word illustrious, James. I'm a lowly electrochemist. So, what we've been doing in my group for 30 years is, using electrochemistry to make nanomaterials like nanowires, and then, finding applications for these things in energy storage and in gas sensing. And, that was my training, back when I was a postdoc. And, Greg and I got together in 2005 and 2006, and what he didn't tell you just now is that we're really celebrating this week because we published a paper on a new type of biosensor just last week that really fulfills the dream that we had, 15 years ago. We had this conversation, we were both really bummed out. And we said, using viruses, we could build disposable sensors that would be cheap and super sensitive, and they wouldn't require cold storage. And, last week we finally, after 16 years of work, we published a paper that really fulfills this dream. And we thought we can adapt this new sensor that we developed to address the challenges of this COVID-19 epidemic. And it's turning out to be true. We're making modifications to this device now, and it looks like it's going to work, and we're really very excited about that. But we're also celebrating this earlier victory on, detecting cancer in urine was really what our dream was.
Why don't you tell us a little bit about that? I mean, that is pretty amazing. Your effort to detect cancer in urine. It's so quick and can facilitate, save lives. It really, promises to save a significant number of lives. Can you give us that story really quickly, if you don't mind, Reg?
Hey, you want to see a picture of our new sensor? Right. I don't know if you can see this, but this is actually two sensors on a glass chip, and the sensor part. Can I try and point to this? I don't even know if this is going to work. But in the middle here, there's a gap between some gold electrodes that you may or may not be able to see. And in that gap, there's a plastic virus composite material that transduces the binding of a cancer marker in urine. If you have bladder cancer, it turns out that you're spilling all of these proteins into your urine every day. And if you could just have a dip and read test for urinalysis that would tell you whether you have these proteins in your urine, then you would know whether you had cancer or were at risk for bladder cancer. And so that's what this little guy does, all right? It detects bladder cancer markers in 1 minute at really, really low concentrations. And that's what we're celebrating this week. And, you know, the path to getting here was tortuous and painful. Isn't that right, Greg?
That's definitely right.
Absolutely. We went down some dead ends, and you know, we published some papers that, you know, looking back, I'm somewhat amazed that we were able to publish some of these papers that we published.
Intermediate steps, Reg.
But the one that we published last week was is truly one of the papers I'm most proud of in my career, I'd have to say.
Thanks, Reg.
If anybody who wants a preprint of this paper, email Greg or I, and we'll be happy to send it to you.
So yeah, that's awesome. And the story is so inspiring. And then now you're able to sort of pivot your expertise and point it towards this new, this sudden and new crisis that we have is also sort of inspiring, and I think brings a lot of hope. John, let me ask your perspective now, just sort of, where are we in, first of all, first round testing, and what role have you guys played in that? Give us give us a little background there, and then now you guys are moving forward with this blood test for the antibodies. So tell us that story, and how is the world reacting, and where are we, and what do we need to do next?
Well, I think there's a debate that you could say, are we testing? Do we have enough tests, right? Is there a shortage of tests? Because when you hear some people in the media, it's like we have all the testing in the world and we're testing more than any other country combined, which may be true, but we're seeing an absolute demand for tests that vastly exceeds the supply. And now there's about 70 companies that have tests that are viral detection tests. So these are the molecular tests that we have. We were one of the first companies. We were like one of probably 5 companies that received the emergency use authorization for our tests. But again, even with all of the alternatives and the vendors that are providing molecular tests, we're each different in little ways. There's differentiation. So our technology, we have an assay design that we feel really, really good about. It's an assay design that's what we call multiplexed. It's 2 different gene targets, 2 different channels. So it takes in consideration any potential mutation of the virus. And that's really important because these things mutate really quick, right? Hence all the different strains of influenza we have. So I would just say that, the first wave was a little bit of a slow start by a lot of us. And I'm culpable in that. I think I told you the story that I grew up in California and I'm used to earthquakes. And every time the building shakes, I don't really pay attention until it really shakes. And then I move out of the house or get underneath the doorway. And that, unfortunately, I take accountability for being maybe a couple weeks late on responding to this, because I've been there with all the other outbreaks. And I knew that we had time to react through big commercial partners like LabCorp or Quest. That was the wrong strategy. We should have moved faster. I apologize to my team for that, and they've made up for it. We have over 200 employees between Northern Italy and Southern California, and we were all on deck every day, every morning, and it was a phenomenal accomplishment. We had emergency use authorization in 30 days from when we said we were going to get into it. So we also had the false start from the CDC. They put out an assay that had problems with, and that didn't help the massive adoption or decentralization of testing as well. So there was a little bit of a hiccup there. Now with this new, our technology detects whether or not you have the virus, right? So it's a molecular test. Now the other question is, have you been exposed to the virus? Do you have antibodies to the virus, right? Have you developed an immunity? And that's what our serology test does. That's a different division of our company based out of Italy. And we're just getting enormous demand for that, for people who want to screen employees before they put them back to work.
So how do you see, that's really interesting. So how do you see, what needs to happen next? You've developed this new antigen technology. It's fully automated. and you're deploying this. What's the kind of next level? And I wonder whether I could get a bit of a back and forth between, say, you and Greg about what, what's the ideal next step for these kind of tests.
Well, I won't steal Greg's thunder because I know where he's going. So I'll just state from our perspective, the maximum throughput is really critical. Maintaining the sensitivity, the specificity, the quality, but getting more throughput so that COVID-19 is screened or tested seasonally with flu. So, we're coming up to a flu season in the fall where our company is now going to offer a test kit that does both. It's not a syndromic panel, but it just does COVID-19, RSV, flu A/B, all together. So you're going to get a seasonal test for both these. It's COVID-19 or the SARS-2 virus is not going away. It's here to stay, in our belief, and that we have to have now very high throughput and very rapid tests to test it.
Yeah, so first, John, congrats to DiaSorin for getting that out so fast. That's awesome. It's exactly what the world needs right now, are tests like those. And I was also really pleased to read in the product brochure that you guys had compared it for neutralizing antibodies. That's critical. That's really the information we need is this going to work against, for the kind of information that people need? Is it going to actually neutralize the virus? The real challenge, though, is, and maybe I can ask you to reflect on this, is, we don't know for now whether those antibodies are going to be maintained and whether they're going to maintain that neutralization. And so how often do you think we're going to end up testing people for this type of thing?
Well, I can only say I hope a lot, right? But
spoken like the business.
That's a loaded question. I'm sorry.
So I'm a huge fan. I like what you guys are doing. I think it's awesome. Let me now pivot and talk to you a little bit about what Reg and I, in collaboration with the company that we co-founded, PhageTech, are working on. So we're developing the next generation type of sensor. So ours is a pretty different approach than what you guys are doing. Reg showed you the electrodes. I think of our test as the Johnny on the spot test. This is the thing that right at the point where you need it, gives you an answer in 1 minute and tells you, do you have these antibodies? Do you have these antibodies? And not just, do you have those antibodies, but how much of those antibodies do you have? So we can start to watch as the antibodies go down or stay maintained. And that's the kind of intelligence that we really need to get to get a real grasp on this and start to choke it and hold it and really reduce its impact on us. So I think all these approaches are great. I'm a big fan. And I think we're going to need a lot of different approaches to get a handle on this thing.
I could probably add one more, what the next steps are as well from, a epidemiological perspective. People are talking about tracing, tracking. So this whole idea of testing, decentralized testing, like Reg and Greg are proposing with a rapid antibody-based test. It's now how quickly can you capture that information electronically, right, or through an app, and be able to inform your physician or your healthcare records of the event that takes place there on being tested. Where were you tested? Who were you around? All these sort of controversial informatic plays coming into effect.
So we've got a couple questions here that are coming in, and I thought I might kind of put them in now because they're relevant to the conversation. There's a couple coming in about the electrochemical phage detector in particular. One of them is, is this going to be so affordable that we could deploy it in all over the world, including third world countries?
So Reg, you go ahead.
Well, the short answer is yes. I mean, one of the design principles of this thing was that it would be disposable and dirt cheap. And part of the reason it can be so cheap is because it's incredibly simple. I mean, it's 2 electrodes and a polymer film. And the receptor in the biosensor are virus particles. And the virus particles do the job of antibodies. Antibodies are very expensive, but virus particles, it turns out, can bind molecules just like antibodies, but they're much, much cheaper to manufacture and more thermally stable as well. And so that's one of the things we're really excited about in terms of this technology, is that these devices could be so cheap.
And let me interject here, Reg, just to mention that this is a different kind of virus, not the coronavirus that everyone's hearing about these days. This is a special kind of virus that's completely harmless to humans. In fact, we've learned to live with it. We've evolved to live with it. It's called a bacteriophage. It only infects the bacteria in your gut, and all of us have it present. So it's something that is totally harmless to us humans and everyone else on the planet, except for the bacteria. It kills the bacteria. So.
And a related question that came in, and I think we know the answer, but you need liquids to do this. I mean, there was a question about whether you could somehow do it in a breath sample using your technology. My guess is you actually need the liquid blood, but correct me if I'm wrong.
That for an antibody test, you would absolutely need some sort of blood. I think John might be able to address air-based monitoring, because you could do that with his.
The viral detection, we're now moving into detecting it in saliva. So we have a swab base right now, both nasal, pharyngeal, so really way up there up the nose, and then nasal, and then bronchial alveoli, BAL. So there's all different types of sample types, but they do involve some type of respiratory liquid.
In theory, though, John, you could simply, have filters that are filtering out air or something, and then dunk those in some buffer, or wash that, right, add it to your test. I bet you guys can have a good market on that. So.
Exactly. Yeah, that's correct.
Oh, that's interesting. So another.
That's the other thing that's in shortage of right now is the transport media, right? So you have all the supply chain issues. You have swabs, transport media. People are coming up with their own customized viral transport media, and this is sort of dangerous because the PCR reactions are all very different. They're all customized. They have like 20 factors that have been optimized all together. You start tweaking salt concentrations, you throw things off, right? So that's an area where we're trying to stay on top of. We have different media types that we have cleared with the assay. We like to have our customers stick to on-label use.
Yeah, didn't that hit here at UCI, James? The lack of viral transfer media?
Yeah, and I think you helped with that, I think. I mean, it was something that we were all trying to do, and the whole campus kind of rallied to produce as much viral transfer media as we could.
Yeah, it was completely nuts. We got this email from the Vice Chancellor for Research suddenly saying, drop everything. This is an urgent request. The medical center is in danger of running out of viral transfer media. And so we ran around the university looking for the materials and put this stuff together.
Wow. Well, listen, we need to buy some from you. Let me get the specs from you afterwards, and we'll do it.
It's honestly, it was stuff we found around, and we were able to make. The university was able to make enough to keep the medical center well supplied. Wow. But yeah, I can definitely help you with that. It wasn't that bad.
I know our platform is being used at the UCI Medical Center, so we have one of our customers as well there.
That's great.
So a question for Reg. So how does your polymer sensor detect COVID antibodies or antigen? So, and then what biofluid are you using? Maybe go over that one more time for everybody.
Well, it's the same trick that we've been using to detect cancer markers in urine, and that is, we deposit a plastic material that contains these viruses that Greg was alluding to, and the viruses once again are harmless, but they have they act like antibodies, and they bind the antibodies that we're trying to detect for COVID-19, and these are the antibodies that are being developed in Greg's laboratory. And then what happens in this polymer film is when that binding event occurs, the polymer film swells ever so slightly. And when that happens, its resistance goes up exponentially, it turns out. And that swelling behavior is fast. It happens in less than a minute. And so that's when we can make a measurement of the concentration of these COVID antibodies, right? And these measurements have to be made in blood serum or whole blood. And right now we're focusing on blood serum, which is a new medium for us. And so there's some challenges in actually working in serum that we're learning about now.
So we've got two questions that I think are both quite interesting, but very distinct, but I think it's worth kind of just going at it. One is, I guess it's more for Reg and Greg, and specifically it's from a chemistry alum who's asking something specific. You know, what are you doing in your labs to ensure that the things are safe? You know, how are we, you know, talk a little bit about the precautions you're taking,
Sure.
that kind of thing.
Sure, yes. And by the way, I noticed that question came in from one of my former student who I think the world of, Dr. Paley. Hi, Miranda, it's good to hear from you. Anyway, this is incredible. Safety is incredibly important to us. Obviously, we're not going to put anyone at risk for anything. So my laboratory has room for about 20 people. We're operating in 2 shifts of 4 students each. So it ensures that people have beyond normal social distancing. And that's really necessary because they're going to spend lots of time breathing in the same, you know, general area. So, and in addition, everyone's required to wear face covering, the CDC mandated face coverings. And then when working with the COVID-19 samples themselves, we're working with those in a biosafety cabinet. There's a special cabinet that can be sterilized and has this laminar flow of air so that you can work safely in there without fear of your own contamination. In addition, they have 2 pairs of gloves, a special barrier coat, safety goggles, face mask, and then I've managed to procure, James, I don't know if I'm allowed to tell you this, but I procured my own N95 face masks that I'm having them wear as well, just to be on the super safe side. The truth is, the risk of that is very low because it would have to be aerosolized. This virus is infectious as an aerosol agent. And so we're, you know, obviously we want to avoid that and just to be on the safe side. So we're doing kind of maximum safety precautions. Nothing comes out of the biosafety cabinet unless it's fully sealed up. And we have a very elaborate, I think we're now at like 20 pages. I think the total amount of paperwork I've contributed to this is probably on the order of about 100 pages or so to get permission to do this and show our protocols and so on and so forth. It's no joke. It's around 100 pages or so of safety requirements that were required to get to this point. Anyway, so Miranda, great question. Thanks.
We have a kind of a broader question now, which I think I'm just going to throw out to you guys. I'm just interested in your thoughts, and maybe we don't know, all of us don't know exactly the answer, but all the states are trying to increase our testing. What are the biggest issues right now? You know, is it test availability? Is it the reagents? Is it sort of lab processing capacity? What are the biggest roadblocks right now?
John, you want to take that?
Yeah, I could say again, from a supply chain perspective, it's a lot of the raw materials that we need to procure to make these tests. You've heard about swab shortages, but again, that's going to be addressed by adding in additional sample types, different like nasal aspirants, for example, is one. I've mentioned saliva. There are, you know, raw materials that, you know, that few vendors actually provide. Let's take the oligos that go in, you synthesize small oligonucleotides as primers, right, for the PCR reaction. There's not many vendors that do that around the world, right? So there's a bottleneck there. But then you have large companies, and I'll give a lot of credit to Thermo Fisher. You know, they're a company that their CEO has been in the White House a number of times. I think he was just in the Rose Garden a couple of days ago, Mark Casper, saying that Thermo Fisher was able to produce something like 5 million tests a week. And that's huge, phenomenal. They're a large company, 75,000 employees, and they're very vertically integrated. But a lot of smaller companies, you know, you're fighting for raw material supply. I think, you know, again, everybody throughout the supply chain are really gearing up and coming to task. So I think this may be as temporary as, you know, some of the scarcity that you're seeing in our grocery stores. Toilet paper, for example. I have no idea why, but that's sort of, as soon as that starts scaling up, we may get more oligos. I don't know.
I mean, that's reassuring. So you're getting the sense now that there's ramp up. People
yeah.
are, yeah.
Absolutely. Yeah. like right now, I could point to our test capacity and tell you that there's no, we're now all the raw material supply chain is in really good control. We've got plenty of supply.
That's really reassuring. We had a quick test, but now back for the polymer test, and I think we know the answer. But the question is the swelling reversible? That is, are these tests reusable? Is it sort of a one-use kind of thing?
The answer is no. Right? They're single use, and let me just explain why that's true. I mean, thermodynamically, if you're going to detect something at 10 to the minus 12 or 10 to the minus 13 or 10 to the minus 14 molar, it has to bind very, very strongly to these virus receptors that Greg is making. And what that means is that the off rate of these molecules that we're detecting is many hours. And so in principle, they will eventually come off the biosensor. Once we've done a measurement, right, we can wash it off, but the washing process takes many hours before all of the stuff that's stuck to our biosensor is off again, and we can make another measurement. And so from a practical standpoint, that's never gonna work. And so, at these concentration levels, we're really talking about single-use devices.
Yeah, let me throw in a little interpretation. I'll be Reg's non-chemistry interpreter here as the biologist on the panel. So what Reg is saying is the viruses grab onto these antibodies and really hang on to them tightly and don't let go on a time frame that's useful for reusing the device. I could tell you this is something of concern to me as just a concerned human. I don't like the idea of throwing out a whole bunch of plastic stuff that's going to get disposed of. So one of the things my lab is exploring is actually printing these. And I believe we might even be able to make it all the way to printing on cardboard or paper or something like that. And that's something we're even exploring now because I want to anticipate what kind of problems we're going to have down the road.
Do you expect to be able to apply this technology more broadly to other kinds of cancers, other kinds of viruses? I mean, how broadly can this be applied?
Absolutely. We're working on bladder cancer now. Reg and I are really passionate about bladder cancer, largely because it comes back so quickly in patients. So we can do a clinical trial very quickly because it just comes back. And we could do our clinical trial of early detection, our dream, within a year or two. And that's really exciting for us. But this should be completely applicable to other cancers, which also involve molecules run amok that appear in urine, that appear in blood, et cetera. And then this opens the door, our COVID-19 test opens the door to applying it to lots of other infectious diseases, things that are also a real big concern, things like malaria, things that are real challenges for us humans on the planet. And we're very excited about that.
So let me ask somewhat maybe more broader for all three of you. Can you walk us through, what is the clinical, what is it like? What hurdles do you have to cross as you move to get sort of an idea, from initial idea to actually sort of helping people out in the real world en masse? Like, what are those steps, and how long does it take usually?
Well, we're on the basic side, so maybe we should turn that one to John.
Yeah,
so he's got a lot more of those than we have.
I give it, so, from idea to patient, so typically it takes us, and I hate to admit this because it varies depending upon the complexity of the test, but, it's almost a 2-year process. And, I told you that we did this in 1 month, and it's just unprecedented, right? Because 6 months of that 2 years is a very in-depth clinical study. Now, this is for a 510K cleared in vitro diagnostic assay. It's it's relatively low risk. It's not like something that you implant that goes through another regulatory process or obviously a drug process as well. So we have a lower threshold. It's the 510K process. Sometimes we've done it much quicker. We've done it from idea to patient in a year, but those were pretty easy assays. The thing that we have for our, in our company and all the companies I've worked for, we have a really detailed product development process. So ideas get curated, they get presented, the business plans get created, and those of you online from industry, you'll know exactly what I'm talking about, a phase gate development process. And at each gate, it's either go, no go. In this case with COVID-19, it was very quick because we got it to the patients under an emergency use authorization. And the federal government, the FDA, has streamlined this to such an extent that our studies were very minimal. We did a limited detection test. We did some safety and efficacy, but really a bare minimum compared to what it would take a typical approval process. So 2 years on average for a typical assay. In this case, it was just a little over a month.
Mm-hmm. Does that concern you at all? I mean, are you?
I get that.
Yeah.
I get that question. I get that concern a lot. Yeah, you have to wonder, are we cutting corners too much? The perception is that we're cutting corners too much. I feel that a lot of this is paperwork. It's a lot of waiting. It's a lot of other projects that are taking priority, right? So even our company, we typically put out 3 to 4 IVD kits a year and many, many more lab developed test components, analyte specific reagents. In this period we're doing COVID, that's all we were doing, right? We put everything else on the back burner. Priority was this COVID assay. And I would expect the FDA is doing the same thing. They're really focused on, all administrative resources on this project. And we were talking to them over the weekends, Saturdays, Sundays, early mornings, late nights. So it's not your typical course of regular business. And but I feel that this is safe for the public. Just for the simple fact that this is technology that's been around for 30 years, and it's a slight variation of what's already existed.
So I'm gonna broaden it a little bit more too, and I'm piggybacking off some questions we're starting to get, and please do feel free to submit questions if people. This is about the time we wanted to open it up anyway. One of the questions that's come up, and I don't think any of you are working specifically on vaccines, but John, you could be. You're doing so many different things.
No, that's out of my league.
But maybe just some thoughts, like the questions that we've gotten several questions. This is like, when is the vaccine happening? Any educated guesses, or are we just waiting? There've been some sort of reasonably good news in some of the reports about tests on monkeys that have come out from the Oxford group, et cetera. You all know more about it than I do, probably. What are your thoughts on a vaccine?
I think it's going to be a long time, to be blunt. At least a year, maybe even a year and a quarter to a year and a half. The issue is you can't just throw this thing out there. You have to make sure that it works, not just on a few humans, but every human. And that's a really high barrier. Plus, there's always a small chance that on some special cases, it will actually make the situation worse, right? It could actually, provoke some sort of unexpected response that then makes the person vulnerable down the road. So anyway, long story short, I'm following this closely. I'm sure, as are most of you, following this very, very closely, watching the Oxford group, for example. And I have some friends at some biotech companies that are working on a vaccine. There's some shortcuts that are being taken that are pretty cool, involving DNA, RNA vaccines, and that's a really cool area. But again, the safety testing, you can't shortcut that. You really have to know that it's going to work, in some very, very high percentage before you roll it out to massive scale. Because remember, when you vaccinate somebody, you're starting with a healthy human. So there's a very low tolerance for something going wrong. And that's going to take a while, unfortunately.
Yeah. I wanted to ask another question, which is just related to the infection rate. We've now seen some reports out, and again, I know I'm going sort of beyond specifically what you guys are working on, but we've got some initial studies in that suggest that based on antibodies, that the infection rate is maybe 50 times higher than the reported cases. We're not testing everyone. What are your thoughts on those results, and how does that relate to the way you're thinking about this problem?
Well, those are really tricky numbers to get around. One of the issues is that the virus has different stages of its cycle. And at some stages, it's shedding out, the patient is shedding out way more virus than others. And so the infection rate could be much, much higher during those stages. And then at other stages, it might be, less virus is getting shed and the immune system is better controlling the virus production. So I think the jury's still out. I think we're still trying to figure that out. I can tell you though, that this is a very, very infectious virus, that this is much more infectious than, just a regular flu, for example. And that's a little bit of a surprise. And it's one of the reasons why we're facing such a big challenge with it.
I think that on the virus detection part, we're using really, really sensitive, specific technology, right? PCR, the gold standard, and the limits of detection are so low. They're like down to the 200 copies per mil. So it's the assay power is there. I think there's more public scrutiny over the sensitivity and specificity of the antibody tests, right? Because there's been companies out there claiming that they could test these antibodies, and there's confusion about how effective these early tests have been. I can't speak to our antibody test specifically, other than maybe we could make the brochure available to everybody that I sent a link to. But again, there's the assay design. In this case, particularly over in the antibody side, is really critical to make sure that you have that performance.
So let me turn now. I mean, we're sort of getting to the sort of 15 minutes before we're going to close out, and I'm going to broaden this even more, if you don't mind. I mean, the world's reaction to this pandemic was mixed. I think a lot of us were sort of hoping that things would work out way better. And, John mentioned that, he's used to the shaking house, and it's only when it really starts shaking do you start paying attention. People have drawn parallels to the world's reaction to the COVID crisis to climate change. This is something, obviously, the COVID crisis happened over months and not, years and years, hundreds of years. And yet we were still slow to react. Do people see parallels there? We've got some deep thinkers on the line here. I wonder if you see parallels in that direction, and what are your reactions?
So I definitely do, James. Thanks for the question, because this is something I'm absolutely passionate about. I see the role of science as not just curing diseases and solving our everyday problems, our biomedical problems, but also making the world a better place, giving us a better way of living. And also anticipating major tragedies and big pandemics like this one. The parallel, though, couldn't be clearer. In both cases, we have experts on the sidelines warning that the train is coming down the tunnel at us. And it was more convenient, I think, to ignore those warnings. So it's really essential for us to get back to listening to scientists and then putting evidence-based rational thinking into policymaking, and really start getting back to what has made our country so successful over the last century or so, which is basically focusing on what are the facts, what does the science tell us, and we're going to go out and do experiments and tell you about that. And in the case of climate change, this is extremely critical, because there will come a point where we can't reverse the effects, and all those years of warning will have been for naught, and that would be beyond a tragedy. That would be an extremely sad situation.
I think that, I think human beings are very data driven, right? We need to see evidence, and some evidence is more apparent to others than others, meaning that, like the COVID example, when you started seeing the curves, right? When you started seeing the death toll, and when you started seeing the aisles in grocery stores that had no product on it, and limiting, one meat product or one bread product per customer, and no toilet paper, no paper towels, no sanitizers, and people wearing masks in, Southern California, it starts becoming, palpable. You start saying, oh, wow, there's something going on here. And then you react. So the reason why I think there was a delayed reaction, you didn't see these visible signs until it was almost too late, right? I think paralleling that to climate change, I think it takes people to really see physical differences in their environment to really wake up and face the, as Al Gore says, right, the inconvenient truth.
Yeah, I mean, I would say that the colleagues that we have and, that I know of in the school and friends of the school who are most passionate about this subject, many of them had their awakenings when they were fortunate enough to, say, visit the Arctic, and you can sort of see with your own eyes that these glaciers are melting. Like, this is no joke. This is real. And you're right, it's visceral. It's probably worth stating, since I put my dean's hat on for a second here. The School of Physical Sciences, we're home to a department, the Earth System Science Department, which is the first of its kind in the United States, devoted to understanding the Earth's climate as a system and humans' impact on that climate. And just a couple of days ago, we had yet another member of that department elected to the National Academy of Sciences, which is the most prestigious scientific body in the world, probably. This is Ellen Druffel, and she studies the carbon cycle and how humanity's burning of fossil fuels affects that cycle, specifically studying ocean carbon. And this is a legacy from a former chair and dean and chancellor Ralph Cicerone, which is, of course, building off the great legacy in atmospheric chemistry here at UCI with Sherry Rowland. So this is a point of significant interest in this school, and in fact, we're going to have another one of these conversations in a couple of weeks, and that will be the subject. We're going to invite experts from Earth Science, Earth System Science, and other places on campus to talk about more of this issue of climate change and COVID-19, how the actual humanity's response to COVID-19 has actually helped things in a minor way in terms of emissions, and you can sort of see what can actually happen if we sort of change output, et cetera. Obviously, we don't want to change output in a way that's going to crash the world economy. That's not the goal here. And we have experts here who work on how you get there while still improving the economy. So sorry for that digression, but I felt the need to jump in and talk about UCI's legacy in this area.
And James, I just want to say that the faculty at UCI feel that legacy very acutely. Our founding chair was Sherry Rowland, who won a Nobel Prize for discovering the reaction that was going to punch holes in the ozone layer. And then most importantly, this is the part that blows me away, that I'm most impressed by. It's not just that he discovered this reaction, it's that he rallied the world to do something about it, right? That he stood up against all these critics who called him all kinds of names, and he convinced people just using his really modest demeanor and just the facts. And he somehow convinced the world to adopt this Montreal Protocol, which has had an incredible impact on our planet and actually turned back what could have been a really dangerous situation. So that's truly inspiring for all of us.
It really is. And you're right, the founding chair of the chemistry department, and it's a legacy I think that's with us. I was chatting with my wife this morning about Roland, and we were just saying, the thing that's amazing about it is you don't lead with the Nobel Prize, you lead with the thing that he did, right? The legacy of the Montreal Protocol and what's happened with CFCs, that's globally important. You don't have to say, and by the way, he won the Nobel Prize for this. That's actually not as impressive as the thing commonly understood. I mean, there are not very many things
that's right
in science that are like that. And
right.
We should be proud of it. And I think we should think of it as a legacy that we can maybe aspire to live up to, all of us.
Absolutely. Yeah. Right. To lay claim to legitimately saving the world, that's amazing. Now, the other thing about Sherry Rowland was he was also one of the nicest, most down-to-earth individuals I've ever met. I would get on the elevator with him and he wanted to talk about baseball or my research, which is amazing. I was an assistant professor when I met this guy. And that legacy carries on. Our department is known for its collegiality. We're known for that kind of down-to-earth, no big egos type of operation. And we really, that comes from the very top. It comes from the guy who saved the world and set that kind of tone in our department. And it's actually really important to us.
Well, I mean, I think I almost feel like I don't want to go on because that's such a wonderfully stated sentiment there. So maybe we should close it out, end on a high note with that. Let me remind everybody, we're going to do this again in a couple of weeks with another group of inspiring and interesting folks to talk about, like I said, more on climate and COVID. And I want to just thank everybody again, everyone who's joined in. Thank you, John. I know you've got a million things going on. You're out there trying to save the world too. So appreciate you taking this hour with us. Really appreciate it. And all your support for the school as well. And Greg and Reg, of course, like this is just fantastic. I appreciate the time you've spent with us today.
Of course, it was a pleasure. Thank you.
Yeah.
All right.
Thanks, James.
See ya. Bye.
So long, everybody.